Our next section is all about the blood and the circulatory system. So, first, we will look at some different types of blood cells histologically. To get an idea of what we're looking at, though, understanding blood is made up of three kind of layers, as we can see. Plasma, which is 55% of whole blood, and plasma contains primarily water, but it also contains proteins, albumin, globulin, fibrinogen, and other plasma proteins. And lastly, all the other good stuff, our electrolytes, gases, vitamins, nutrients, hormones. Anytime you're collecting a blood sample, and it says it came from the serum or the plasma, that is where we're measuring it from, is kind of that area, that section, which appears yellow here. The buffy coat is the separation layer. This is the white blood cells, leukocytes, and platelets, and it's less than 1% of whole blood. It's a really small fraction. And then lastly is the hematocrit, or the red blood cells, the erythrocytes, which is about 45%-ish of whole blood, although there is an AMAB-AFAB distinction. You can see here in this image, we have normal blood listed, anemia, where you'll see the hematocrit is depressed, or polycythemia, where the hematocrit is elevated. When we look then at what we can see histologically, there are the formed elements in the blood, and these are the cells. So our thrombocytes, which is our platelets, our leukocytes, which will break down into granulocytes and agranulocytes. And then lastly, what we see primarily on this picture, these red things, these are our red blood cells, or our erythrocytes. How we are able to view these under the microscope is by creating a blood smear, which looks somewhat like this, where you essentially get a drop of blood on the slide, and you take one other glass slide and you drag the end until it reaches the blood, and it creates a thin seal, and then you push that blood across the slide, creating this streak. And this flame kind of tail region is where we look at our blood cells, our whole blood under the microscope, and you can add some stains to it as well. Looking at our first formed element, our erythrocytes, or red blood cells, they contain hemoglobin for oxygen and carbon dioxide transport in the blood. They have a biconcave shape. They do not contain a nucleus or organelles, and they stain kind of eosinophilic. They appear pink under microscopy. They are the most abundant cell type in the whole blood, so they're going to be the majority of the thing that you see when looking at peripheral smears, and they live 100 to 120 days. A reminder of what hemoglobin is. Remember, it's a globulin protein, or globin protein. It's created by 4 polypeptide chains that contain heme molecules that bind iron in the center. Now, our red blood cells don't always look that perfectly circular shape and size. There are pathologies, one of note called sickle cell disease, where we can see that the red blood cells, instead of being circular and kind of biconcave shape, they are sickled in nature and almost look like a crescent. And we can see several of them there in this image. In sickle cell anemia, or even sickle cell disease, these cells can essentially clump together, especially in small capillaries, small blood vessels, and then get stuck and plug these blood vessels, causing ischemia and microvascular injury and damage. Hereditary spherocytosis is another condition where, instead of seeing a perfectly kind of spherical red blood cell, we can see a red blood cell that is not really maintaining its normal shape and structure. Our erythrocyte shape is normally maintained by cytoskeletal elements that help provide mechanical stability and also flexibility, but that sometimes can be shifted or changed with certain genetic conditions or other conditions. So hereditary spherocytosis, we see that our red blood cell is not a biconcave disc anymore, but it's actually a sphere. So it looks almost like a globe, and it would be protruding out against us. It's lost to that kind of collapsed shape in the center. And another pathology connection is microcytic hypochromic anemia. And we see this here with this really kind of pale red blood cell that we're seeing compared to the ones that we've seen before. And iron deficiency anemia is an example of a microcytic hypochromic anemia, because we're able to classify red blood cells by size and color. So a normocytic red blood cell is a normal sized red blood cell. A small red blood cell is considered microcytic, and a large red blood cell is considered macrocytic. And so with a microcytic anemia, we have a small red blood cell, and we don't have enough of them. You can also classify red blood cells on their coloration. Are they well They kind of nice and pink, that would be normochromic, or are they hypochromic, meaning that they likely don't have enough hemoglobin or potentially enough iron, as in iron deficiency anemia. So this is an important distinction that we start doing when we move into pathology. Our next formed element are our leukocytes, which are our immune system cells that make up less than 1% of blood volume. They move through the body and exit at our post-capillary venules to enter into tissue. They are highly motile cells, and they are signaled towards these areas of inflammation or infection via chemokines, which are chemicals. There are two categories of leukocytes, granulocytes and agranulocytes. Granulocytes have lysosomes and granules, and they include things like neutrophils, eosinophils, and basophils. Agranulocytes do not contain granules, they just have lysosomes, and they would be like monocytes or lymphocytes. Speaking of a monocyte, they travel through blood to enter into our tissue, and inside the tissue, they transform into a macrophage or even another type of specialized phagocytic cell like an osteoclast. They are the largest white blood cell, and they have a large, slightly indented nucleus that looks like a kidney bean, as seen in these images. There are no prominent granules in the cytoplasm. It makes up to about 5 to 10% of white blood cells in a peripheral white blood cell differential, and they can live in for days as monocytes in the blood, and they'll live longer actually in tissue as macrophages. A macrophage is seen here in this image. These are phagocytic cells that will ingest and engulf foreign substances and cells. They then can also present these antigens using MHC major histocompatibility 2 molecules. Have an oval or a slightly indented nucleus, and oftentimes their cytoplasm looks foamy after they've ingested stuff, they phagocytosed stuff, but they can be challenging to distinguish, and we often don't see these in the blood. A lymphocyte, seen here, can travel from the blood into connective tissue and lymph. They are not just one type of cell, there are many types of cells. So T cells, B cells, and natural killer cells all would be lymphocytes. They are smaller or similar size to red blood cells, and they have a really dark spherical nucleus that has a slight indent to it. Then surrounding, they have a very thin rim of cytoplasm. They are about 20 to 40% of white blood cells. There's lots more even in tissue, and they can live up to years, but maybe even as short as days, depending on the subtype. Now, plasma cells are a subtype of a lymphocyte, and these are actually a subtype of a B cell or a B cell lymphocyte, and these live in connective tissue. And these cells secrete antibodies, and these are immunoglobulins, and those antibodies tag antigens for destruction. They have a clock face nucleus because they have peripheral heterochromatin. They have a very basophilic, dark staining cytoplasm. And you can see this lighter area, that's a visible Golgi apparatus or visual Golgi image. Our next cell type is our neutrophils. These are the first wave of cells that move towards infection or towards acute inflammation in tissues. They can phagocytose. They also contain lysosomes and granules that fuse with their phagosome and digest the material. Many and most neutrophils will die during this process, and they will produce or form pus. These cells are larger than red blood cells, and they have a multi-lobed nucleus, as seen here. Have poorly staining granules in the cytoplasm, and they make up to 40 to 60% of white blood cells by percentage. They'll live only for a few hours in the blood and just a few days in tissue. Our next cell type is eosinophils. These migrate into tissues, and they play a major role in defense against a parasitic infection. They also can attenuate an allergic reaction, and they're found oftentimes in our gut, in the lamina propria of the gut, as well as our respiratory tract. They are larger than a red blood cell, and they have a bilobed nucleus. They have lysosomes and eosinophilic staining granules in the cytoplasm, and there tends to not be many of them, 1 to 5% of white blood cells, and they'll live for a few days in blood and tissues. Our basophils can mediate inflammatory reactions, and they can release two substances, heparin, which acts as an anticoagulant, and histamine, which acts as a vasodilator. They are similar in nature to mast cells, and they're larger than our red blood cells. They contain lysosomes as well as basophilic granules that stain kind of blue, and these granules obscure a bilobed nucleus, as seen here. They're very rare, less than 1% of white blood cells, and they'll live for a few days in blood and tissues. As I mentioned, a similar cell is mast cells, which live in the connective tissue. They're like basophils. They mediate our inflammatory response and can release histamine and heparin, and they have granules and lysosomes that are present in the cytoplasm. Lastly, we have our tiny little platelets. Our platelets, or thrombocytes, are essentially part of our clotting cascade or blood clotting. They create a platelet plug. They're much smaller than red blood cells. When a blood vessel is injured, the exposed underlying connective tissue will come in contact with platelets, and then that activates them. They then release granules that mediate aggregation and adhesion, which is blood clotting. And they have a variable cytoplasm with no nucleus. They are very abundant and they only live a few days in the blood. And while this is a really small picture to look at, we can zoom in here on this image and see that this contains all of our different cell types and kind of walks through the amount that you should see and kind of their claim to fame. When we come back, we're going to talk about different blood vessel types.